40,000 years ago, a mammoth named Yuka died in the Siberian ice, and now, against every rule of biology, her living genetic messages have been read. Scientists recovered not just DNA, but fragile RNA, revealing which genes were still firing as she froze, something no fossil has ever preserved. What they found is not just rewriting textbooks, it is threatening our understanding of extinction, disease, and even resurrection. But how did these living genes survive the impossible? And what secrets are buried in the molecular diary Yuka left behind? RNA is the genetic world's drama queen, famous for being fragile, fleeting, and frankly, a little high maintenance. Biologists have always said DNA is the tough guy, able to survive for tens of thousands, sometimes even a million years if you keep it cold and dark. RNA, on the other hand, is more like a souffle. Blink and it collapses. Under normal conditions, RNA starts breaking itself down within hours of an animal's death. Even in a laboratory freezer, the best-case scenario is a few years before it turns to mush. The accepted record for RNA survival was under 100 years, and that was in specimens carefully preserved by humans, not something left outside for 40,000 years. So how did any of Yuka's RNA make it through the Ice Age gauntlet? The answer lies in Siberia's permafrost, which is less like a freezer and more like a time capsule. When Yuka died, her body was buried almost instantly in a deep freeze, locking out oxygen and bacteria, the usual suspects in the crime of molecular destruction. The temperature never climbed above zero, not even for a day. There were no thaw cycles, no spring mud, no hungry scavengers getting a taste. It was as if nature herself slapped a do not disturb sign on the mammoth and left her on ice for the next 40,000 years. But even with this perfect cold storage, the odds were still laughable. RNA is chemically unstable by design. It is supposed to deliver genetic messages and then self-destruct, like a secret agent who eats the evidence. The idea that any of these molecules could last long enough for modern scientists to read them is, to put it mildly, the molecular equivalent of finding a working flip phone in a volcano. That is why the research team's first reaction was not excitement but suspicion. If RNA from Yuka really survived, it would mean rewriting the basic rules of biology. And in science, rewriting the rules means you need proof. Lots of it from every angle, and then some. The next step was not celebration, it was a forensic investigation. Inside Stockholm University's ancient biomolecule facility, the gloves were on, the air was filtered, and the skepticism was thick enough to cut with a pipette. Emilio Marmol Sanchez and Love Dalen led the team through a protocol that read more like a crime lab checklist than a biology experiment. Every surface was UV sterilized, every tool tracked by lot number, and every sample entered the clean room, double-bagged, logged, and handled one at a time. Extraction blanks ran alongside every batch. If a single stray molecule of modern RNA showed up, the whole process stopped, and the benches got a bleach bath. The first results landed with all the subtlety of a dropped beaker. From Yuka's muscle, liver, and skin, the sequencers called out nearly 300 distinct messenger RNA molecules, each a genetic message frozen since the Ice Age. Alongside those, about 60 microRNA molecules surfaced, tiny regulators that act like post-it notes on the genome, marking which genes should be read and when. The numbers were not just high for a fossil, they were higher than some fresh museum specimens. Negative controls came back clean. Fragmentation patterns matched what you would expect after 40 millennia in the deep freeze, short, battered, and unmistakably real. Mark Friedlander and the bioinformatics team double-checked every read, comparing them to both modern elephants and known lab contaminants. The sequences did not match any modern animal, nor did they show the telltale signs of lab mishaps. Instead, they mapped to extinct mammoth genes with unique mutations that sealed their authenticity. The team's excitement was measured, almost clinical, as if afraid the molecules might evaporate if anyone cheered too loudly. But the raw data was undeniable. For the first time, a fossil had yielded not just ancient DNA, but the living instructions, the actual gene activity preserved from the moment of death. 40,000 years of scientific dogma had just been outvoted by a few hundred stubborn molecules. Before anyone dared to publish, the team put their findings through the scientific equivalent of a lie detector. The first stop was Copenhagen. An independent lab received tissue samples under strict cold chain custody, with paperwork tracking every handler and every minute out of the freezer. Scientists there ran the same extraction and sequencing, using their own reagents, their own machines, and, just for fun, their own set of extraction blanks. If contamination had snuck in anywhere along the chain, it would have shown up as modern RNA in these controls. Instead, the blanks stayed blank. The Copenhagen sequencers turned up the same battered short fragments, matching Stockholm's results almost letter for letter, right down to the unique chemical scars left by 40,000 years of deep freeze. Meanwhile, back in Stockholm, bioinformaticians combed through every read for telltale damage signatures. Ancient RNA does not just look old. It falls apart in a very specific way, with chemical nicks and base changes that modern molecules never show. These patterns match what is seen in the oldest DNA fossils, and nothing like the tidy, intact RNA you would get from a modern elephant or a stray sneeze in the lab. Every step was logged, every anomaly flagged. When a single batch showed a hint of modern contamination, the team trashed the reagents, sterilized the benches, and started over. No shortcuts, no wishful thinking. All of it was verified. The real test came when the Copenhagen team emailed their results late one night. The message said the same gene fragments, the same patterns, the same impossible story. For a moment, the group chat froze. Then came a flurry of all-caps messages, a few reactions, and finally a scientist's version of a victory lap, with people double-checking the negative controls one more time. That independent replication changed everything, with independent replication and contamination controls holding up under scrutiny. Disbelief gave way to celebration. The data was real, and so was the breakthrough. Only then did the team allow themselves to wonder what those ancient genes were actually saying. Gene by gene, Yuka's final hours come into focus, not as a story told in bones or teeth, but as a molecular record written in panic and cold. The RNA sequencers picked up a surge of cold shock protein transcripts, the kind cells crank out when temperatures plummet and survival instincts take over. These proteins, including CIRBP and RBM3 analogs, act like the genome's emergency blankets, working overtime to keep cell machinery from freezing solid. Their presence, still detectable in battered fragments, is a chemical fingerprint of hypothermia. Alongside the cold response, another pattern emerges. Metabolic shutdown genes, the cellular equivalent of rationing supplies, show Yuka's body was running on empty. Glucose transporter RNAs and fat metabolism markers are all lit up, evidence that the mammoth was burning through its last reserves. This is not just the story of being cold, it is the story of starvation. The same RNA profile appears in modern elephants facing famine, but here it is locked in time, unchanged, for 40 millennia. Stress response transcripts run like a red thread through the data. Elevated signals from hormone regulators and heat shock proteins point to a body in crisis, flooded with chemical signals of alarm. In the gut, fragments of plant RNA linger, remnants of the last meal, blades of Ice Age grass digested but not forgotten. For scientists, this is not just a list of gene names. It is a molecular autopsy, a reconstruction of Yuka's struggle against cold, hunger, and fear, preserved in the language of her own cells. A new layer of suspense crept in when the team began analyzing fragments that did not belong to the mammoth at all. Buried among Yuka's gene transcripts were pieces of ancient viral RNA, genetic shrapnel from Ice Age pathogens that vanished tens of thousands of years ago. Some fragments matched extinct influenza strains, and others looked suspiciously like coronavirus relatives. These were not complete blueprints for a pandemic, but they were enough to make even the most stoic lab techs pause before lunch. The risk was not about resurrecting a mammoth version of the flu. These sequences were shattered, incomplete, and about as likely to reassemble into a living virus as a jigsaw puzzle dropped down a garbage disposal. Still, the fact that viral RNA could survive alongside mammoth genes was a wake-up call. If permafrost can preserve the genetic fingerprints of ancient diseases, what else might be thawing out as the Arctic melts? That is the kind of question that keeps biosecurity officers up at night and makes science fiction writers giddy. Before the findings went public, the team debated how much to reveal. Some argued for holding back the viral data, worried it might inspire a new genre of do-it-yourself virology. Others insisted that transparency was the safest path, reasoning that sunlight and bleach are the best disinfectants. In the end, the publication included the pathogenic fragments, accompanied by several pages of biosafety guidance. When asked if they had just handed the world a recipe for disaster, one researcher shrugged and said, "Only if you are planning to build a pandemic out of broken Legos." Cold resistance genes from Yuka's frozen cells are now a hot topic in medical research circles. The same cold shock proteins her body churned out in its final hours, molecules like CRBP and RBM3, are already being studied for their ability to protect human tissues from freezing damage. Emergency doctors and trauma surgeons are looking at these ancient blueprints, wondering if they might help improve outcomes for patients with hypothermia or those undergoing organ transplants. Instead of guessing how to shield cells from the cold, researchers now have a working playbook written by an animal that survived Ice Age winters, right up until it did not. The lessons do not stop at temperature. The battered RNA fragments from Yuka's tissue also offer clues about how mammals respond to starvation, stress, and even viral threats. Some of the gene activity patterns match what is seen in modern elephants during drought or famine, providing a living reference for wildlife biologists and veterinarians. And those shattered viral sequences, while no threat to public health, are a goldmine for pandemic preparedness. By studying how Ice Age viruses interacted with their hosts, scientists can model how new pathogens might jump between species or adapt to extreme environments. The hope is that these ancient genetic snapshots could someday help design vaccines or treatments before the next big outbreak has a chance to spread. For all the talk of de-extinction, the real revolution may be in medicine and disease control. Yuka's RNA is not just a relic, it is a toolkit for solving problems that are still very much alive. Venture capitalists don't usually line up for Ice Age biology, but once Mammoth RNA hit the journals, the phone lines lit up. The reason is not just nostalgia for furry elephants. It is because RNA gives researchers something DNA never could: a real-time map of how an extinct animal actually functioned. DNA is the full parts list, but RNA is the instruction manual, showing when to build thick fur, when to store fat, and when to crank up cold resistance. For the first time, scientists can see which genes were switched on in a freezing mammoth, and when those survival pathways kicked in. That is not just trivia, it is the missing piece for anyone hoping to engineer a living, breathing mammoth. With these gene activity patterns, biotechnologists can start modeling the timing and strength of traits like wool density or subcutaneous fat, instead of just guessing from skeletons and hair clumps. That is a game changer for de-extinction projects. Instead of blindly inserting mammoth DNA into elephant cells and hoping for the best, now there is a blueprint for which switches to flip and when. It is no surprise that startups and university labs are quietly licensing this data, betting that the next breakthrough will come from getting gene expression right, not just the raw code. The promise is big, but even the most optimistic researchers are careful to point out, knowing the instructions is one thing, building the machine is another. The mechanics of resurrection are still a work in progress. The discovery of ancient RNA from a frozen mammoth set off a frenzy of speculation. Paleontologists in Montana's Hell Creek Formation have uncovered T-Rex fossils with traces of soft tissue, and some people wonder if dinosaur RNA could be within reach. The chemistry delivers a harsher verdict. RNA is fragile even in the best conditions, and 66 million years is a marathon no molecule is built to survive. Even the most optimistic researchers admit that, outside the deep freeze of permafrost, the odds of finding readable dinosaur RNA are vanishingly small. Mineralization replaces living tissue with stone, leaving no safe harbor for the molecular machinery of life. The debate does not stop with ancient animals. Russian collections include Neanderthal remains preserved in permafrost, and that raises the possibility of recovering RNA from extinct human relatives. Here, the conversation shifts from scientific curiosity to an ethical minefield. Recovering Neanderthal gene activity could unlock secrets about human evolution, disease, and adaptation. But questions of consent, dignity, and the rights of the dead rise quickly. Many countries have strict rules or outright bans on extracting genetic material from ancient humans, so researchers tread carefully to avoid crossing ethical lines. The excitement of discovery is always checked by the responsibility to respect what and who we study. While Mammoth RNA opens new frontiers, it also draws a bright line around what science can and should attempt. Arctic permafrost has always been a slow-moving clock, ticking away beneath the surface while the rest of the world barely notices. But now that clock is running out of time. Over the last 2 decades, Siberia has seen record-breaking heat waves, temperatures climbing high enough to turn what should be solid ground into rivers of mud. In 2021 alone, satellite data showed over 100,000 square miles of permafrost began to thaw for the first time in millennia. With every summer, millions of ancient specimens, mammoths, cave lions, even bacteria, are being exposed and lost before anyone can study them. Scientists estimate that up to 6 million mammoth remains are still locked in the Siberian ground. But with the thaw accelerating, the window for discovery is closing fast. This is not just a race against the elements. It is a question of who decides what happens next. Right now, there is no international agreement on how to manage these genetic time capsules. Some call for global oversight, arguing that the world's oldest DNA and RNA should be protected as a shared resource. Others push back, saying local communities and nations should decide, not distant committees. Meanwhile, commercial interests are circling, eager to license or even privatize the next big find. The responsibility is not just in the hands of scientists anymore. Whether these frozen archives become tools for research, medicine, or something else entirely depends on choices made today, by policymakers, by communities, and by anyone who cares about what's left beneath the ice. Today, ancient genes thaw as fast as the Arctic melts. With each degree of warming, irreplaceable genetic evidence vanishes, along with clues to evolution, disease, and resilience. The clock is running. Scientists estimate Siberian permafrost could lose most preserved specimens within decades. What we choose to recover now does not just shape our past, it could decide our biological future. What would you risk to read the next frozen chapter?